Hello, my name is Leslie Jimison, and I'm from the Department of Bioelectronics at the Center of Microelectronic de Provence of the EMSE. I hope English is okay. Today I'm going to talk about the use of organic electronics for applications in biosensing. So I work in the Department of Bioelectronics, and when I refer to bioelectronics, I'm talking about the coupling of biology and electronics. This involves the translation of biological signals to signals we can read with traditional electronics, and vice versa, using traditional electronics to read and control biological systems and signaling. There are a number of bioelectronic applications already on the market are glucose monitors that provide on-the-site diagnostics. The rest of these devices range from early prototypes to clinical trials. Smart aid bandages or smart bandages are being developed to monitor wound healing. Ocular implantations can provide artificial vision. And at the Penn Center for Brain Injury and Repair, artificial limbs are being tested that are controlled by the brain. Now, organic electronics refers to devices that use carbon-based polymers and small molecules as the active material. They have a number of advantages, including their easy processing and low-cost fabrication, tunability of electronic properties, ionic and electronic conductivity, ideal surfaces, and soft mechanical properties. Their soft nature and compatibility with soft substrates makes them ideal for things like flexible displays and large solid-state lighting. It also makes them ideal for interfacing with biological systems. We think that organic materials will lead to new applications in bioelectronics. So one subject we are focusing heavily on in our group is organic bioelectronics for pathogen detection and diagnostics. So particularly, we're interfacing with various biorecognition elements, and this includes enzymes, antibodies, and pathogens. And we are paying special attention to pathogens. I define a pathogen as any infectious biological agent causing disease to its host. This can include viruses, fungi, bacteria, protozoa, and parasites. Pathogen detection is very important, both for individual health, food safety, and drug development. Current methods of pathogen detection include cell culture, which is shown below, PCR, immunoassays, biochemical test, and microscopy. When designing new methods for pathogen detection, it's important to take into consideration speed, sensitivity, specificity, and cost. And we believe we can design an in vitro model for pathogen detection based on the organic electrochemical transistor, and this is a technology I'll introduce in a couple of slides. So we're developing this device to detect the integrity of barrier tissue. Barrier tissue comprises tight layers of cells that form a barrier to block passage of pathogens and toxins, but allow the passage of ions and nutrients. Examples in the body include the gastrointestinal tract and the blood-brain barrier. This cartoon to the right shows a layer of tightly packed epithelial cells. Transport of ions can occur in either two pathways: paracellular transport between the cells, or transcellular transport within the cells. Protein complexes, known as tight junctions, exist between the cells to regulate paracellular transport. And in the first part of this project, we're going to be focusing on this paracellular transport and the function of the tight junctions. And the second part of the project, we'll be focusing on transcellular transport, which is controlled by ion channels in the cell membrane. So there's a number of ways that we can measure barrier and tissue integrity in our lab. This includes permeability assays, where we tag a molecule with a fluorescent marker and watch how easily it passes through the cell membrane. We can also directly image using immunofluorescence. You've seen this a couple of times today. We also have a tool called the Celsiscope, which with two electrodes on either side of the cell layer can measure the electrical resistance. This is a very useful tool for monitoring barrier formation over time. So we want to use the organic electrochemical transistor in order to measure this barrier tissue integrity. So I want to take a moment to go over the mechanics of the organic electrochemical transistor. Key parts include a gold or metallic source and drain contact, a conducting polymer channel that's in contact with an electrolyte, and submersed in this electrolyte is a gate electrode. The conducting polymer system we use is PEDOT-PSS, and in this system, negative charges exist along the PSS background. These are fixed. The negative charges are compensated by positive holes along the PEDOT polymer chain. These positive holes are very mobile, and in the presence of the field or a voltage drop across the source and drain, they easily move. In this state, the film is considered doped and is highly conducting. Now, if we put a positive charge on this gate electrode that's immersed in the electrolyte, we're encouraging positive ions to penetrate into the conducting polymer film. In order to maintain charge neutrality, therefore, less positive holes are needed to compensate these negative charges. These large positive ions are less mobile. This means in this state, the film is considered de-doped and is less conducting. So with the same electric field across the source and drain, we get a lower source drain current. This de-doping and doping doesn't happen instantaneously, but we can monitor this characteristic rise time. When we put a square voltage pulse on the gate electrode, we can watch what happens to the drain current. So how do we use this to monitor cells? We have this organic electrochemical transistor here, and in between the polymer channel and the gate electrode, we culture a layer of cells on a porous cell culture membrane. After 3 weeks of culturing, they form a tight monolayer. Tight junction proteins are shown here in green and the cell nucleus in blue. When this layer is intact, when these cells are healthy, they form a barrier to ion transport. We get a slower de-doping in the device. If we then damage these tight junctions, or these tight junctions aren't present, there is no ionic barrier to transport between the gate electrode and the polymer channel. De-doping is faster, device response time is faster. So in order to mimic a pathogen attack, we use hydrogen peroxide. It's been shown before that hydrogen peroxide causes damage to the cell membranes, with the damage corresponding to hydrogen peroxide concentration. Shown here are healthy cells compared to cells exposed to 20 millimolar and 100 millimolar hydrogen peroxide. On exposure to the hydrogen peroxide, the tight junction proteins are less organized. We have also shown in our lab with permeability assays that compared to a healthy control, the addition of hydrogen peroxide greatly increases flux and apparent permeability. So we can also show this same effect with our OECT. This plot shows the drain current when we have a healthy cell layer, shown in blue, and then we have no cell layer, shown in black. When no cell layer is present, the device response time is faster. We get an overall lower modulation. The presence of healthy cells suppresses this modulation, so there's less current change between the on and the off state. The great thing about our device is we can watch this effect in situ. We apply periodic square pulses to the gate electrode. In this case, 1 second on, and then we wait a full minute. So each one of these pulses is a minute. We add 60 millimolar of hydrogen peroxide, and we immediately see a change. This is a fast effect and a fast measurement system. This plot better shows the comparison between the healthy cell layer response and then when the cells are exposed to hydrogen peroxide. We see that although the hydrogen peroxide is not completely destroying the cell layer, we are destroying the ion barrier, or the barrier for transport of ions. And so in summary of this project, we have shown or demonstrated that the effect of pathogen mimicking substances, such as hydrogen peroxide, can be observed in situ. We were pulsing for once a minute, but we can pulse a lot faster. This is one of the quicker characterization methods for pathogen detection. The OECD response can be modeled to give more quantitative values of barrier tissue integrity, such as the transepithelial resistance, which is a common figure of merit to compare epithelial and endothelial cells. Our future work will focus on device optimization, paying particular attention to sensitivity, and then we'll start to answer more biologically relevant questions, such as what happens when we add different concentrations of hydrogen peroxides or expose the cells to real toxins. We believe this OECT can be a powerful tool for the development of in vitro models for barrier tissue, drug screening, and toxicology. So I want to close with a little bit broader look at the Department of Bioelectronics at CMP. We aim to be an internationally renowned department in which bioelectronic technologies are generated through a better understanding of the electronics and tissue interface. We want to provide an outstanding education opportunity in this rapidly evolving field. So we do this by forming an intimate integration of materials, electronics, and life sciences. We have a large number of non-permanent rotating personnel that are experts in these various disciplines. We have key alliances with teams throughout the world, such as Cornell and Stanford. We also participate in the organization of international conferences, including the Materials Research Society, the European Materials Research Society, and Gordon Research Conferences. Our research at a glance comprises 3 aims. I've already talked a little bit about our diagnostic tools. We are also interested in the interface between neuroscience and bioelectronics, and using organic electronics to control cell adhesion. So shown here is a highly conformable array of PDOT electrodes and organic electrochemical transistors. We use photolithography to define side-by-side PDOT electrodes and the PDOT transistors. This ECOG array is then placed on the surface of the brain of a rat, and alongside a state-of-the-art silicon probe is also implanted. We can see from the signaling here that the PDOT electrode senses the same events as a silicon probe. This data was taken during an epileptic seizure. Moreover, the PDOT transistor also senses the same signaling with a very impressive signal-to-noise ratio. This is the first time that such neuron activity has been recorded in the brain with a transistor. And we're very excited about this, and there's a lot of ongoing work. If you want to hear more about these projects too, we have a poster by Sebastian Sinar, my colleague. In regard to cell adhesion, we have shown before that we can pattern a conducting polymer stripe and put a voltage gradient across of it. This controls the oxidation state, producing an oxidation gradient, and influences cell adhesion. Cells tend to prefer the oxidized side. We now know that this is because the oxidation state is in fact controlling conformation of surface proteins. In particular, we looked at the conformation of fibronectin. And fibronectin is a protein very important for cell adhesion. We can show that on one side of the voltage or oxidation gradient, the fibronectin is compact, whereas on the other side, it is open and available to the cell. Moreover, this shows that we can uniformly control protein conformation across a large length scale, which is not very easy to do. Moreover, we can directly correlate the protein conformation with cell migration, which is shown in these plots. When the protein is compact, the cells tend to move and do so in this downhill direction towards the other side of the gradient. And when the fibronectin is open and available to the cells adhere better and there's less migration. So in conclusion, the Department of Bioelectronics was created at CMP of the EMSE in 2009. We're a relatively young department, but we're growing quickly. We explore the fundamentals and applications of coupling electronics with biology, and we have a core strength in the use of organic electronic materials. We have 3 faculty. George Malieras acts as full professor and department head, and associate professors we have Roisin Owens and Sebastian Sinar. We have a number of PhDs and postdocs like myself, and a number of very important collaborations. I'd like to thank all of our funding, including the ERC, which sponsored the project I talked about at the beginning of the presentation. And I'd like to thank you for your time and open up the floor to any questions.
Thank you very much. We have plenty of time for questions and answers, probably. So who is interested in organic transistors and applications? Yes?
I have just a question about the fibronectin. Normally, fibronectin is trapped in collagen, etc., etc. How you can measure what you measured? Did you measure the fibronectin cleavage in a matrix or in
No, the
matrix?
fibronectin was exposed to the conducting polymer surface after the conducting polymer was deposited, and then we use fluorescence resonance energy transfer to monitor the conformation of the proteins on top. So not exactly what you would see maybe in the body, but I think we understand what's going on well.
Okay, thank you.
Does that answer your question?
Yeah, yeah, it's perfect.
More questions? I was just curious to know how far are we from application to human beings with these organic transistors? Do you have a guess for that?
I can't tell you.
No? No guess?
Not tomorrow.
Not tomorrow, yes. The day after tomorrow?
Yeah, I have a related question about these signals to detect ion transfer through a layer of cells, if I understood well. First of all, a 60 millimolar H2O2 is quite a strong,
Yes.
very strong, unspecific oxidative stress for cells. So I have no doubt that you might detect something, and I'm wondering whether or not this has any physiological meaning. And the second, I mean, with respect to future application to human, and if the idea is to detect different types of pathogens. First of all, not all pathogens will have an effect on tight junctions, and to which extent you will have some specificity in the detection, given that major infection will anyway, whatever the pathogen is, will provide some oxidative stress and will maybe, to a certain extent, give the type of effects you observed. But how are you going to say, is it a virus? Is it a bacteria? What type of pathogen? So I'm quite skeptic.
Yeah, so there's a lot of
That
parts.
level.
There's a lot of parts of the question, so remind me if I don't answer one of them. I'll first tell you that this is the very early stages of this sensor. And so the first goal was just to see that we see a change when we have a tight barrier and when we don't. 60 millimolar is high, but we're using it in media, and so some of the proteins we believe quench the hydrogen, the effect of hydrogen. What's not shown in this presentation is we can sense at least 5 millimolar and probably a lot smaller. This is not our detection limit. Hydrogen peroxide was used first only to mimic the attack on tight junctions. Our next step is to look at more reversible effects to make sure we're not just destroying everything, that we're actually just touching the tight junctions and then removing that stress. We've done other controls that show that the response of the organic transistor is not due to the hydrogen peroxide alone, so we have that taken care of. As far as specificity of the device, right now we're still exploring what we can do, but our key advantage of this device is the temporal resolution. And if there's a way that we could correlate time signatures of viruses or pathogens with their with the attack that we see, that could be very useful. We won't be able to sense pathogens that aren't attacking the tight junctions. This device is being used to understand pathogen attack on tight junctions. So we are limited, but we also have our strengths.
Other questions? You just mentioned briefly the activity of the Microelectronics Center of Gardanne. Are all activity related to biological applications, or do you have?
No,
yeah?
there's 3 departments, and Sebastian, correct me if I'm wrong, but there's a department of bioelectronics, which is where I work, and then there's there's two other departments to deal more with the organic electronics, and then the third one is informatics. Flexible security. Yes. So, not all bioelectronics, sorry.
Organic electronics is important. Is an important topic. Okay. Well, no, I'm wrong.
Bonjour, je me présente, Sébastien Sanor, je suis maître assistant dans le département de bioélectronique du CMP à Gardanne. Donc il y a 4 départements de recherche sur le CMP, le site de Gardanne. Donc le département de bioélectronique dont on a eu l'exposé, dont la majeure partie des travaux de recherche se basent donc sur l'électronique organique. Il y a un département de packaging et support souple, un département de système architecture sécurisée pour les domaines d'application de la carte à puce, et un département de sciences de la fabrication et logistique.
Merci pour cette précision. Alors, est-ce qu'il y a une toute dernière question? Bon, ok, il n'y en a pas. Ok, thank you very much. Nous allons passer à la table ronde.